Synchrotron-based X-ray fluorescence is an important technique for observing elemental segregation, stoichiometric relationships, and clustering behavior in samples from a multitude of fields, including biology, chemistry, and material science. The information obtained from these studies is qualitative until proper quantification procedures are used to convert raw fluorescence counts into elemental areal masses. This video will demonstrate how to use the quantification program created by Argonne National Laboratory to generate numerical information for two-dimensional X-ray fluorescence maps. To use the MAPS program, it is first necessary to download the IDL software from the internet. This can currently be done by going to the website for IDL and creating an account. Next, select My Account, then Downloads, and it will show a page of all the available programs. Scroll down and select the most recent version of IDL. Next, maps may be downloaded from the Argonne National Laboratory website. After downloading and extracting the zip folder, there should be 4 files: compound.dat, hanky.xdr, maps, and xrflibrary.csv. The 3 files other than maps should be copied and pasted in the IDL subfolder called lib. For Windows computers, this most likely can be found within Program Files under the folder Excelis. Generally, it is convenient to run the fitting from the desktop. However, it is critical that the folder name and the pathway do not contain any spaces or special characters. Otherwise, maps will produce an error when attempting to run the fitting. If the pathway to the desktop does contain spaces, place the folder elsewhere, for example, directly within the C drive. For this demonstration, I will place the fitting folder and the maps.sav icon on the desktop for easy access. Within this folder, place the files maps fit parameters override.txt.